Dear students, in the present lecture, we shall try to learn the mechanism of a light reaction, which is otherwise called a photochemical phase. In the previous lecture, we have learned the mechanism of photosynthesis, how light reaction and dark reaction are interrelated with each other, and how the site of reaction and all. In the present lecture, we shall learn about the light reaction of photosynthesis. See, as we have learned in the previous lecture, that is, light reaction is a light dependent reaction. It was discovered by Robert Hill, hence it is called Hill's reaction. And it is also called as a photochemical reaction because light energy is converted into chemical energy. Hence it is called as a photochemical phase or a photochemical reaction. And this takes place in the thylakoid membrane of the grana region of the chloroplast. And the raw materials required to bring about light reaction are pigments, that is photosystem 1 and photosystem 2, water and sunlight. We have two raw materials for the photosynthesis to happen. It is one is carbon dioxide, other one is water. So in the first phase of the mechanism, that is in the light reaction, only the water makes its entry into the mechanism, and sunlight is of course required because it is a light dependent reaction. And to absorb light energy, what are required are pigments are required. What are these pigments? These are the quantasomes, photosystem unit 1 and photosystem unit 2, present in the membrane of thylakoid of the grana region of the chloroplast. These three are required. And the light reaction consists of the three main phases. One is called as the photo excitation of chlorophyll A molecule, and photolysis of water, and photophosphorylation. In under these three headings, we can learn the mechanism of light reaction. If you consider the first one, that is photo excitation of chlorophyll A molecule, means to say the all the photon of light which travels from the solar sunlight in the form of photon bundle of light rays hits the chlorophyll molecule, and all the pigments, if it is ps1 250 and if it is ps2 300, all the pigments molecules in the chlorophyll absorb light energy. After absorbing light energy, the chlorophyll molecule gets excited. Why it gets excited? Because it gets extra energy from the solar, that is solar energy. So after getting extra energy, it's get excited, and one of the valency electron in the valency orbital shift to the next higher energy level. One of the valence electron shift to the next energy level. It is because of extra energy gained by this chlorophyll molecule. And the chlorophyll, now referred as a chlorophyll present in excited state. Why it is excited? Because it has extra energy, and it is proved by showing its electron at excited level. So this will remain in excited state for a period of 10 to the power of minus 8 seconds. And again, the same chlorophyll A molecule, which has gained extra energy, now loses energy and again comes back to ground state. And that extra energy gained will be utilized in converting the light energy into chemical energy. That is why it is called as a photochemical reaction. So how light energy is converted into chemical energy? The all the pigments absorbs light energy in the form of photon, and the chlorophyll now gets extra energy, moves to excited level, and it remains in the excited level for a period of 10 to the power of minus 8 seconds. And during that period, 10 to the power of minus 8 seconds, whatever the amount of light energy that is received by the chlorophyll molecule will all be converted into chemical energy. After the lapse of 10 to the power of minus 8 seconds, the chlorophyll, which is in the excited state, now comes back to its ground state. Why it comes back to its ground state? Because it has lost its energy. in converting light energy into chemical energy. That is why it is a light reaction is also called as photochemical reaction, where light energy is converted into chemical energy. Now, the next step that is called as photolysis of water and evolution of oxygen. The name itself indicates that photo means light, lysis means breakdown of water. The water is broken down, that is lysis, breakdown of water into H plus ions and OH minus ions, hydrogen ions and hydroxyl ions. In presence of light energy is called photolysis. Photo means light, lysis means breakdown. Breaking down of water into H plus and OH minus ions in presence of light energy is called as photolysis of water, for which manganese, calcium and chloride plays a very important role for splitting up of water. So photolysis of water can also be defined as splitting up of water molecule into H plus ions and OH minus ions in presence of light is what is called as photolysis of water. The OH minus ions thus formed during photolysis, see in the second reaction, recombines together forming water, two molecules of water, and oxygen is liberated. Look at the arrow mark. Oxygen is liberated. This is the oxygen which is liberated at the end of the photosynthesis as a byproduct. So this experiment, that is photolysis of water experiment, clearly proves that the water which is liberated at the end of photosynthesis as a byproduct comes from water. The oxygen which is liberated at the end of the photosynthesis as a byproduct comes from water, but not from carbon dioxide. Why? It is because carbon dioxide still has not made its entry into the reaction mechanism. It's only the water which has entered. So this oxygen is liberated from the water. So this is what is called as photolysis of water. So this oxygen is a byproduct. See, before the formation of product, that is glucose, a byproduct is formed during photosynthesis. And these H plus ions, 4 H plus ions, are accepted by the hydrogen acceptor NADP to form NADPH2. That is reduction reaction. Photosynthesis is called as redox process, reduction and oxidation. So addition of hydrogen is a reduction. So there are many NADP molecules, nicotinamide adenine dinucleotide phosphates, where they react with 4H plus ions forming NADPH plus H plus, that is reducing agent is formed. Now the next step is the photophosphorylation. The name itself indicates that photo, light reaction, phosphorus, relation, addition of inorganic phosphorus to an ADP to form ATP during the light reaction of the photosynthesis is called as photophosphorylation. Photophosphorylation is a process of synthesis of ATP from adenosine diphosphate and PI, inorganic phosphate, during light reaction. It is known as photophosphorylation and it was discovered by Arnon in the year 1954. See, the photophosphorylation takes place in two different stages. One is cyclic photophosphorylation, other one is called as a non-cyclic photophosphorylation. In both cases, ATP is formed. The reactants for formation of ATP are only the ADP and IP, inorganic phosphate. So one is formed during the cyclic journey of the electron, that is called cyclic photophosphorylation. The other ATP is formed during non-cyclic journey of the electron, that is called non-cyclic photophosphorylation. So if you consider the first one, cyclic photophosphorylation, the process of synthesis of energy-rich compound ATP utilizing ADP, inorganic adenosine diphosphate and inorganic phosphate during the cyclic journey of the electron, which is expelled from the excited chlorophyll A molecule, is called as cyclic photophosphorylation. This could be explained by this cycle. This is the schematic representation of a cyclic photophosphorylation. See here, cyclic photophosphorylation requires P700, that is photosystem unit 1, where photosystem unit 1 contains 250 pigment molecules. Out of 250 pigment molecules, all the pigments absorb light energy. Look at these arrow marks. These are the photons of light. All the 250 pigments absorb light energy, but The conversion of light energy into chemical energy is brought about by only chlorophyll A 700 or P700. Hence, P700 is said to be what is called as a reaction center. So this chlorophyll A molecule, after getting the light energy in the form of photon, it gets excited. As I told earlier, the excited chlorophyll loses an valence electron. Electron present in the outermost orbital gets expelled from the excited chlorophyll A. This electron passes through various electron acceptors like FRS, ferredoxin reducing substance, PQ, plastoquinone, cytochrome B, CYTB, and cytochrome F, and plastocyanin. Again, it comes back to the chlorophyll A. So this chlorophyll A 700, after getting extra energy, that is photon of energy from the photon, it gets energy rich, excited, and one of the valence electron is expelled, and this electron passes through various electron acceptors like ferredoxin reducing substance, plastoquinone, cytochrome B, cytochrome F, plastocyanin, and again it ends up with a journey in chlorophyll A 700 only. Now, this chlorophyll A700, after getting the same electron which was expelled, now comes back to its ground state. During this cyclic journey of the electron, when the electron passes between the ferredoxin reducing substance and plastoquinone, there is a phosphorylation, that is addition of inorganic phosphate to adenosine diphosphate ADP, ending up with the formation of ATP. Here is the arrow mark. Look at the arrow mark reaction. So an ATP is formed from ADP and IP. This is called as formation of ATP from ADP and IP is called as photophosphorylation. This photophosphorylation is taking place during the cyclic journey of the electron. Hence it is called as cyclic photophosphorylation. So one of the ATP is formed when the electron is between FRS and plastoquinone, and another ATP is formed by one more photophosphorylation process when the electron passes between cytochrome B6 and cytochrome F. So ultimately, two ATP are formed during cyclic photophosphorylation. During cyclic photophosphorylation, and look at the this is the explanation. Non-cyclic photophosphorylation, another method. See, here we can explain with this. This is the schematic representation of a non-cyclic photophosphorylation, where the very difference between cyclic photophosphorylation and non-cyclic photophosphorylation is that during the cyclic photophosphorylation, there is a cyclic journey of the electron, number one. During a non-cyclic journey of the electron, this one, non-cyclic journey of the electron, look at here, the journey of the electron is not cyclic. Hence it is called as non-cyclic journey or non-cyclic photophosphorylation. It is also called Z scheme electron transfer because it somewhat looks like a Z. The electron transfer process somewhat looks like a Z pattern. Hence it is also called as Z scheme electron transfer. And another notable difference between cyclic and non-cyclic photophosphorylation is in cyclic photophosphorylation, there was only the involvement of P700. And here in non-cyclic photophosphorylation, we have both P700, PS1 and P680, PS2. So involvement of two photosystems in non-cyclic and only one photosystem in cyclic. And here only one ATP is formed during non-cyclic photophosphorylation, whereas in cyclic photophosphorylation, two ATP were formed. And here NADPH2 reducing agent is formed. In cyclic photophosphorylation, there is no formation of reducing agent. And we have also here photolysis of water. Water gets oxidized. Splitting up of water into H plus OH minus ions takes place during non-cyclic, whereas during cyclic photophosphorylation, photolysis of water does not take place. So this during non-cyclic photophosphorylation, where the same P700, that is photosystem unit 1, gets light energy in the form of photons. When it gets light energy, it gets excited. After absorbing light energy, it gets excited, goes to an excited level. When it goes to an excited level, one of the valency electron of the chlorophyll A gets expelled from this excited chlorophyll A molecule P700, and this electron passes through electron acceptor called ferredoxin reducing substance, and later from ferredoxin reducing substance to ferredoxin, and again the electrons are accepted by NADP, and NADP gets reduced to form NADPH2. Look at the arrow mark. This is nicotinamide adenine dinucleotide phosphate. This is nicotinamide adenine dinucleotide phosphate reduced. Reduction of NADP is formed to form NADP plus H plus. And here the electron which is expelled from the PS1, excited chlorophyll A, PS1, started its journey from PS1 and ended up with the formation of NADPH2. There is no cyclic journey. Even the PS1 still in excited state. At the same time, there is entry of another chlorophyll A molecule called as photosystem 2, that is where the reaction center is P680. Even this chlorophyll is supplied with energy after getting energy in the form of photon. Look at the arrow bundle of light rays. After getting energy, even this P680, this PS2 gets excited, goes to an excited level. When it goes to an excited level, even this also expels one of its electrons, the valency orbital, and this electron passes through various electron acceptors, as you can see here. Plastoquinone, cytochrome B6, cytochrome F, plastocyanine. Likewise, it passes through various electron acceptors, and the electron of the P680 ends up its journey after passing through various electron acceptors, ends up its journey with P700. P700. After getting the electron, the P700 now, which was in excited state, now comes to a ground state. It it was it was in excited state after losing an electron, and thus that electron has ended up its journey with NADP, forming a reducing substance NADPH2, and still it was in excited state. At the same time, the P680 enters the scheme where after getting energy, a photon of light energy in the form of photons, even this P680 expels one of its electrons from the valence orbital, and this electron passes through various electron acceptors, ending up with the journey with ending its journey with P700, bringing P700 from excited state to a ground state. During this process, when electron of the P680 passes between cytochrome B6 and cytochrome F, a phosphorylation is formed, an ATP is formed from ADP and IP. This phosphorylation is called as photophosphorylation, and since this photophosphorylation takes place during non-cyclic journey of the electron, it is referred as a non-cyclic photophosphorylation. Non-cyclic photophosphorylation. At the same time, the here the NADP electrons are accepted. Electrons of P700 passes through FRS and FD and accepted by NADP. How it's get reduced? For the reduction of NADP, it requires hydrogen. So here it comes. Water molecules are there. Splitting up of water molecules into H plus and OH minus ions is called photolysis of water. That takes place during light reaction. So these H plus, I told you in the previous slide, these H plus ions are accepted by NADP molecules, forming NADPH2. That is, formation of reducing agent NADPH2 is formed. So at the end of light reaction, what are all formed? Say, conclusion. At the end of the light reaction, assimilatory powers like NADPH2 formed during non-cyclic photophosphorylation, ATP, two ATP molecules during light reaction, cyclic photophosphorylation, and one ATP during non-cyclic photophosphorylation are formed along with the liberation of oxygen. So the end products of light reaction are two molecules of NADPH2, three molecules of ATP, and oxygen are formed at the end of a light reaction. So in general, light reaction is a light dependent stage of the photosynthesis, which takes place in the grana region of the chloroplast, where the thylakoids are present in presence of the pigment systems and the water in presence of sunlight. So here light reaction takes place in three stages: photolysis, photoexcitation of chlorophyll A, conversion of light energy into chemical energy, photolysis of water, then third is formation of reducing agent NADPH2, fourth is photophosphorylation, that is formation of ATP, bicyclic and non-cyclic photophosphorylation. Thus, at the end of the mechanism of light dependent reaction, light reaction, that is Hill reaction, assimilatory product like NADPH2, three ATP molecules and an oxygen are formed at the end of light reaction. Now, these assimilatory products like NADPH2 and ATP enters the chemical energy, which is light energy, which is preserved here in NADPH2 and ATP, now enters the Calvin cycle, where Calvin cycle, that is dark reaction, is a light independent reaction. It takes place in the darkness, in the absence of light, where by utilizing the indirect form of light, stored light energy in NADPH2 and ATP. So in the next lecture, we shall try to learn about the mechanism of dark reaction, that is Calvin cycle.